Hello everyone, welcome to ITLS Academy. This is the second part of the mechanism of action of antibiotics. So let's start. First is chromosome function and replication. Chromosome function and replication. The basis for selective inhibition of chromosome replication and function. देखते हैं. As with protein synthesis, the mechanism of chromosome replication and functions are essentially the same in prokaryotes and eukaryotes. There are, however, important differences in the detailed functioning and properties of the enzymes involved, and these differences are exploited by a number of agents as the basis of selective inhibition. The microbial chromosome is large in comparison with the shell that contains it. During replication, the circular double helical must be unwound to allow the DNA polymerase enzyme to synthesize new complementary strand. Here, the shape of the chromosome is manipulated by the shell by the formation of regions of supercoiling. Positive supercoiling makes the chromosome more compact. Negative supercoiling produces localized strand separation, which is required for replication and transcription. In the bacterium such as E. coli, 4 different topoisomerase enzymes are responsible for maintaining the shape of DNA during cell division. They act by cutting one or both of the DNA strands. They remove or generate supercoiling, then reseal the strands. The basic sequence of events for microbial chromosome replication is as follows. First is synthesis of precursors. Purines, pyrimidines and their nucleosides and nucleoside triphosphate are synthesized in the cytoplasm. At this stage, the antifolate drugs, that is, sulfonamides and dihydrofolate reductase inhibitors, act by blocking the production of thymine. The antifungal agent, that is 5-fluorocytosine, interferes with these early stages of DNA synthesis. Through conversion to 5-fluorouracil, then to 5-fluorodeoxyuridylic acid, it blocks thymidyllic acid production through inhibition of the enzyme thymidylate synthetase. The antiviral nucleoside acycloguanosine, that is acyclovir, and idodeoxyuridine, that is idoxyuridine, are converted to their respective nucleoside triphosphates in the cytoplasm of infected cells. They proceed to inhibit viral DNA replication either by inhibition of the DNA polymerase or by incorporation into DNA with subsequent termination of chain extension. Finally, the anti-human immunodeficiency virus, that is HIV drug, is azithromycin. Azithromycin acts in an analogous manner, being converted to the corresponding triphosphate and inhibiting viral RNA synthesis by the HIV reverse transcriptase. Next is unwinding of the chromosome. DNA double helix must unwind to allow access of the polymerase enzyme to produce two new strands of DNA. This is facilitated by DNA gyrase, that is the target of the quinolones. Some agents interfere with the unwinding of the chromosome by physical obstruction. These include the acridine dye, of which the topical antiseptic proflavin is the most familiar, and the anti-malarial acridine, mepacrine, etc. They prevent strand separation by insertion or intercalation between base pairs from each strand, but exhibit very poor selective toxicity. Next is replication of DNA strands. The unwound DNA strands are kept unfolded during replication by binding a protein called Albert's protein. A series of enzymes produce new strands of DNA using each of the separated strands as templates. An RNA polymerase forms short primer strands of RNA on each template strand at specific initiatal sites. DNA polymerase third then synthesizes and joins short DNA strands onto the RNA primers. These DNA strands are called Okazaki fragments. DNA polymerase 1, which possesses nucleotidase activity, removes the RNA primer and replaces them with DNA strands. Finally, a DNA ligase joins together the DNA strands, producing two daughter chromosomes. The entire process is carefully regulated with proofreading stages to check that each nucleotide is correctly incorporated as specified by the template sequence. There are no therapeutic agents yet known which interfere directly with the DNA polymerase. Next process is transcription. The process of transcription, that is the copying of a single strand of mRNA sequence using one strand of the chromosome as a template is carried out by RNA polymerase. RNA polymerase is a complex of four proteins, that is 2 alpha, 1 beta and 1 beta prime subunits. This RNA polymerase complex made of 2 alpha subunits, 1 beta and 1 beta prime. These are subunits of RNA polymerases which make up the core enzyme. Another small protein, the C factor, joins the core enzyme which binds to the promoter region of the DNA preceding the gene which is to be transcribed. The correct positioning and orientation of the polymerase is obtained by recognition of specific marker sites on the DNA at position -10 and -35 nucleotide bases before the initiation site for transcription. The A factor is responsible for recognition of the initiation signal for transcription, and the core enzyme possesses the activity to join the nucleotides in the sequence specified by the genes. Mammalian genes possess an analogous RNA polymerase, but there are sufficient differences in structure to permit selective inhibition of the microbial enzyme by the semi-synthetic rifamycin antibiotic and rifampicin. Next is quinolones. The quinolones selectively inhibit DNA gyrase, that is topoisomerase II, which is not found in mammalian cells. The gyrase, a tetramer comprising of 2 alpha and 2 beta subunits, is a highly versatile enzyme which is capable of catalyzing a variety of changes in DNA topology. These include introduction of negative supercoiling and removal of positive supercoiling. Removal of positive supercoiling is the process of relaxation. Apart from these also work in unknotting and removal of linked structure, that is decatenation. Such activities ensure that the daughter chromosomes produced during replication can segregate in the cytoplasm prior to cell division. The gyrase binds to the chromosome at a point where two separate double-stranded regions cross. This can be at a supercoiled region, a knotted or a linked or catenated region. The A subunit or alpha subunit cut both DNA strands on one chain, while a 4 base stagger the other chain is passed through the break, which is then released. The beta subunits derive energy for the reaction by hydrolysis of adenosine triphosphate. This is the process of segregation of this is the process which is carried out during replication Although we are going to cover this all, that is fundamental processes for molecular biology, in the lecture series of molecular biology in the category of CSIR and GRF life sciences. If you want, then you can join and you can study the whole molecular biology there. Next point is the action of the quinolones probably triggers secondary responses in the cells which are responsible for death. One notable morphological effect of quinolone treatment of a gram-negative rod-shaped organism is the formation of filaments. Some of the quinolones may also act upon topoisomerase 4, which appears to be more important for chromosome segregation in Staphylococci. Nitroimidazoles and nitrofurans. Nitroimidazoles are metronidazole and nitrofurans are nitrofuranotoin. These agents also cause DNA strand breakage, but by a direct chemical action rather than by inhibition of a topoisomerase. Metronidazole is active only against anaerobic organisms. The nitro group of metronidazole is converted to a nitronate radical by the low redox potential within cells. The activated metronidazole then attacks the DNA, producing strand breakage. Nitrofurantoin is thought to act in a similar manner. Next is our rifampicin. The action of rifampicin is upon the beta subunit of RNA polymerase. Binding of just one molecule of rifampicin inhibits the initiation stage of transcription, in which the first nucleotide is incorporated into the RNA chain. Once started, transcription itself is not inhibited. It has been suggested that the structure of rifampicin resembles that of two adenosine nucleotides in RNA. This may form the basis of the binding of the antibiotic to the J3 subunit. These changes of rapid development of resistance in organism due to alteration in the amino acids do not affect the activity of the polymerase but render it insensitive to rifampicin. The action of rifampicin is specific for the microbial RNA polymerase, the mammalian version being unaffected. Next is 5-fluorocytosine. This antifungal agent inhibits DNA synthesis at the early stages involving production of the nucleotide thymidyllic acid, that is DTMP. 5-fluorocytosine, that is 5-FC, is converted by a deaminase inside fungi to 5-fluorouracil. Here 5FC is converted to 5-fluorouracil. Then to the corresponding nucleoside phosphate, that is 5-fluorodeoxyuracilic acid, or we can say 5-FTUMP. This compound then activates or acts as an inhibitor of thymidylate synthetase, which normally produces 1-DTMP from uridine monophosphate by addition of methyl group supplied by a folate cofactor to the 5' position of the uracil ring. As this position is blocked by the fluoro group, that is 5-FDUMP acts as an inhibitor to the enzyme. 5-fluorocytosine can be considered as a prodrug. It has the value of being taken up by fungi as the pyrimidine base, whereas the active metabolite produced inside the cell would not be taken up because of its negative charge. Next is folate antagonist. Folate metabolism in microbial and mammalian cells. Folic acid is an important cofactor in all living cells, that is, it is present in all living cells. In the reduced form, tetrahydrofolate or THF, it functions as a carrier of single carbon fragment which are used in the synthesis of adenine, guanine, thymine and methionine. One important folate dependent enzyme is thymidylate synthetase, which produces DTMP by transfer of the methyl group from tetrahydrofolate to DUMP. In this and other folate dependent reactions, tetrahydrofolate is converted to dihydrofolic acid, which must be reduced back to tetrahydrofolate before it can participate again as a carbon fragment carrier. There is another fundamental difference between folate utilization in microbial and mammalian cells. Bacteria and protozoa are unable to take up exogenous folate and must synthesize it themselves. Bacteria and protozoa synthesize their own folate. This is carried out in a series of reactions involving first the synthesis of dihydropteroic acid from one molecule each of pteridine and paminobenzoic acid, that is PABA. Glutamic acid is then added to form dihydrofolate, which is reduced by dihydrofolate reductase to tetrahydrofolate. Sulfonamides. Sulfonamides are structural analogues of PABA. They competitively inhibit the incorporation of PABA, that is paminobenzoic acid. They competitively inhibit the incorporation of PABA into dihydropteroic acid, and there is some evidence for their incorporation into false folate analogues which inhibit subsequent metabolism. The presence of excess PABA will reverse the inhibitory action of sulfonamides. Here the excess PABA has the inhibitory effect on the action of sulfonamide, as will thymine, adenine, guanine and methionine. However, these nutrients are not normally available at the site of infection, for which the sulfonamides are used. Next is our dihydrofolate reductase inhibitors. Trimethoprime is a selective inhibitor of bacterial dihydrofolate reductase. The bacterial enzyme is several thousand times more sensitive than the mammalian enzymes. Pyrimethamine, likewise, is a selective inhibitor of plasmodial dihydrofolate reductase. Here, these both are structural analogues of the dihydropteroic acid portion of the dihydrofolate substrate. Crystal structures of the bacterial, plasmodial and mammalian dihydrofolate reductases, each containing either bound substrate or the inhibitors, have been determined by X-ray diffraction studies. These show how inhibitors fit tightly into the active sites normally occupied by the dihydrofolate substrate, forming a pattern of strong hydrogen bonds with amino acid residues and water molecules lining the site. The cytoplasmic membrane. Composition and susceptibility of membranes to selective disruption. The integrity of cytoplasmic membrane is vital for the normal functioning of all cells. As we know that bacterial membranes do not contain sterols, and in this respect these bacterial membranes differ from membranes of fungi and mammalian cells. We can say that fungal membranes contain predominantly ergasterol as the sterol components, whereas mammalian cells contain cholesterol, while The sterols are absent in bacteria. Gram-negative bacteria contain an additional outer membrane structure which provides a protective penetration barrier to potentially harmful substances, including many antibiotics. The cytoplasmic membrane of gram-negative bacteria have an additional outer membrane that is known as LPS membrane, or we can say lipopolysaccharide membrane. This provides a barrier for the penetration of harmful substances even the dyes. As we know, in the gram staining, there is a purple color obtained in gram positive bacteria, while there is no color, or we can say after the washing from the alcohol, there is a removal of the color from the gram negative bacteria. This is just because of the additional outer membrane, that is lipopolysaccharide membrane, present in the gram negative bacteria. Outer membrane has an unusual asymmetric structure in which phospholipids occupy the inner face and the lipopolysaccharides occupies the outer face. So the outer membrane is attached to the peptidoglycan by proteins and lipoproteins. The stability of all membranes is maintained by a combination of non-covalent interactions. These non-covalent interactions include hydrogen bonds, hydrophobic bonds, ionic bonds, van der Waals interaction, etc. between the constituents involving ionic, hydrophobic, and hydrogen bonding. Polymyxins. Polymyxin B and polymyxin E, that is colistin, are used in the treatment of serious gram-negative bacterial infections, particularly those caused by Pseudomonas aeruginosa. We can say that polymyxin is an important antibiotic in inhibiting the infection of Pseudomonas aeruginosa. They comprise a cyclic peptide containing positively charged groups linked by a tripeptide to a hydrophobic branched chain fatty acid. The outer leaflet of the membrane structure is distorted, segments of which are released and the permeability barrier destroyed. This is the action of antibiotic, that is polymyxin. Polymyxin distorts the membrane structure. Apart from this, the polymyxins can then penetrate to the cytoplasmic membrane where they disrupt membrane integrity, causing leakage of the cytoplasmic components. Next is naphthidine. Naphthidine, this synthetic allyl amine derivative, inhibits the enzyme squalene epoxide at an early stage in fungal sterol biosynthesis. Acting as a structural analog of squalene, naphthidine causes the accumulation of this unstructured hydrocarbon and a decrease in ergosterol in the fungal cell membrane. Next is polyenes, amphotericin B and nystatin. These are the most commonly used members of this group of antifungal agents, that is polyenes. They derive their action from their strong affinity towards sterols, particularly ergosterols. The hydrophobic polyene region binds to the hydrophobic sterol ring system within fungal membranes. In so doing, the hydroxylated portion of the polyene is, is pulled into the membrane interior, destabilizing the structure and causing the leakage of cytoplasmic constituents. Next is imidazoles and triazoles. The azole antifungal drugs act by inhibiting the synthesis of the sterol components of the fungal membrane. We can say that they are inhibitors of one step in the complex pathway of ergosterol synthesis involving the removal of a methyl group from lanosterol. The 14A demethylase enzyme responsible is dependent upon cytochrome P450. The imidazoles and triazoles cause rapid defects in fungal membrane integrity due to reduced level of ergosterol with loss of cytoplasmic constituents, leading to similar effects to the polyenes. The azoles are not entirely specific for fungal ergosterol synthesis and have some action upon mammalian steroid metabolism. For example, they reduce testosterone synthesis. So thank you very much guys for watching this session. Here the mechanism of action of antibiotics is completed. This was the second part of the mechanism of action of antibiotics. We have studied various antibiotics that are responsible for inhibiting the action or the for inhibiting the various actions, the like replication, transcription and translation etc in the body. So we have completed the mechanism of action of antibiotics. Next we will continue with our another lecture. We will continue with next lecture in this series. So thank you very much for watching this session. You can subscribe ITLS Academy on YouTube. You can also get the app of ITLS Academy on the Google Play Store. So stay tuned with ITLS Academy, and we'll meet you soon in the next lecture. Thank you very much for watching this particular session.